Yo, welcome back to JC Audits. We have a amazing speaker for you today. His name is Archie, and he's studied some kind of neurobiology, if I've got that right. I'm sure you'll find him interesting. I'm gonna spike him with a few questions and see how he does. Um, I've got all my faith in him. I think he'll do really well, and hopefully he'll be coming online shortly. And yeah, just all this about prions and what is a prion, and if you like, I'm going to try and nail him down on what is a virus as well, even though he's not supposed to be talking about viruses. But yeah, see if he has anything to say about that, because obviously we've had our fair share of viruses. It's now 2023. Of course, we get seasonal flu every winter. Plus, 4 years ago, we had COVID, didn't we? So, is it SARS COVID coronavirus 2? Um, yeah, COVID is a virus, as we all know. But what is a virus? Um, is it is it an RNA virus? Is it a DNA virus? Are they encapsulated in some way? Protein coat? Um, Maybe you can sort of like go over that, even though he's not supposed to talk about that. And definitely, what I'd like to hear what he's got to say about prions. Um, yeah, hopefully he's gonna come online shortly, and we're gonna chat to him about that. Let's have a look. So, what I wonder what your questions would be to Archie about that, and even this is a pre-recorded video. I'm sure Archie would not mind answering a few questions on prions if you wanna, or maybe on viruses as well. If you wanna drop in a question in the chat, either when this goes live or even a few months after this goes live. We're currently in March 2023, so feel free to drop a note, and I'll try and get Archie to come up with some reply. So I'm just gonna pause the video for now and see if I can get Archie in the room. So I'll see you in one moment. Yeah, so should we just? I'll just say like a short introduction. Then I've already done a little introduction on my channel, but maybe you can correct me, because I'm bound to have a couple of mistakes. First, in your little bio, what I can remember, and then secondly, before you launch into your stuff, I just wanted to get a word in, just in case I might not be able to in the next few minutes. But all I want to say is I'm so grateful for you to give up your time to be on this channel. That's number one. Number two, from what I understand, what you studied was something to do with the brain and biology. I might call you a neurobiologist, but I know you're going to correct that in a minute. And the next thing is, maybe at some point, I know you're not supposed to talk about this, you were going to talk about prions, but maybe you could also draw a parallel between prions and viruses and what the differences might be. I'm going to shut up now. You've got the floor.
Super. Yeah. So I'm Archie. I'm an analyst for the NHS. I studied psychology at Bangor University as an undergraduate, and then I decided to move into more biology-related areas. So I went to Bristol University and studied molecular neuroscience as a master's, which kind of sounds quite fancy, but it's essentially just brain chemistry, brain biochemistry. still don't really know how it worked properly, and it's going to be a very long time before we have a firm understanding of how it works. I realised I wasn't very good at lab work, so I moved out of the sort of the wet lab work, so working in a lab, pipetting liquids into tubes and using centrifuges and stuff, and moved more into the sort of data analysis side, so statistics and research methodology a little bit, but that's not really within my scope anymore. So I mean, neurobiology is sort of right. Neurobiology might relate more to a sort of theoretical sort of like exploratory science role, whereas this is a lot more treatment focused, sort of disease and treatment focused. So I was going to talk about prions. Prions are not much is actually known about them. You ask anyone in the street what a prion is, they might be able to tell you that it's a disease, but other than that, I don't think many people would really know what a prion is, and actually how scary they are. Really, they are quite frightening. So yeah, I mean, it all started back in the 50s. There's an uncontacted tribe, I believe, in Papua New Guinea, and the women and children were getting these bizarre symptoms of sort of becoming incapacitated and having fits of laughter that they couldn't control, and then they were sort of dying soon after, and they had these kind of like symptoms that mimic dementia, essentially. And this group of researchers go over there and they do some tests, and they realised that it was only the women and the children that were getting it, and it didn't take long for them to notice some sort of quite odd behaviour, like for instance, this particular group of people would engage in sort of ritualistic cannibalism, so they would eat their dead, not necessarily kill people to eat them. They would eat like members of their group who had died naturally or died prematurely, and the women and the children they had the privilege of eating the brain of the person who died. The whole event, all of the events that lead up to the researchers realising that's what's causing it is it's a very long story. We'll be here for ages if I explain all of that. But these researchers, they hypothesised that it was because they were eating the brains, and so there was like a campaign to work out how and why this was happening. And fast forward to the 90s, you had something called mad cow disease, which a lot of people in this country who are sort of like probably 30s or above will remember the mad cow disease. It was causing cows to behave bizarrely, and hence why it's called mad cow disease. And then they were dying, and they suspected that it was being leaked into the meat that people were eating, and the connection between these two is they're both prion diseases. So in the earlier disease where the people were eating the brains, the disease is now called Kuru, spelled Kuru, and it's a type of prion disease that's caused by eating the brains of the people. They suspect that in the early 1900s there was a patient zero who had something called CJD, or Creutzfeldt-Jakob disease, which is what you'd get if you ate beef from a cow that had mad cow disease. So it's mad cow disease, but for humans. So it's a prion disease, and it's called CJD, or Creutzfeldt-Jakob disease. and the symptoms are pretty dire. They're fatal every single time. So once you get CJD, then it's a death sentence. And so it does mimic dementia in most ways. So there's a lot of neurological symptoms. There's a lot of cognitive symptoms. There's a lot of sort of like wasting. I don't know if you know anyone who's had dementia or who has dementia. It's a very sort of wasting disease, wasting of your personality, your character, your memories. There's also like physical symptoms that can come along with it as well, usually due to like the changes in behaviour, but I suspect there's also biological correlates to it as well. So yeah, with CJD, they reckon that this patient 0 had this CJD, and then the brain was eaten, and this is where the whole like infectious part of prions come in. So prions are stands for proteinaceous infectious particle. So it's not a virus, it's not a fungus, it's not a bacteria, it's not I forget what the fourth one is. It's like malaria. It's a bit like a parasite, but it's not quite a parasite. But it's not a parasite. It's not the same thing as what malaria is. Plasmoid. No.
Okay.
Anyway, it's unimportant. It's none of those things. It turns out that prions are not even alive at all. They're proteins, misfolded proteins. So, are you aware of what a protein is and what it's made up of?
I'm guessing it's made up of amino acids and.
Yeah. So you've got this these endogenous amino acids that are created in animals and humans. 26, I believe, amino acids that are commonly used in humans, and so the prion, this prion in humans and other animals as well, is I think it's just over 200 amino acids long, and we have an endogenous version within us inside of us that sort of like is important for cells. It's called PRP. But the misfolded version that causes CJD, it has a slight change in the way it's folded, so it's a slightly different shape to the normal version. And when this protein comes into contact with other versions of itself, but different versions, then it causes those versions to become the same way as this sort of diseased state, and then they stick together. And then they go and find more, and they confirm that they change the conformation of those proteins so that they're the wrong shape. Then they join them, and this process happens over and over and over again until you get these plaques, these big clumps of proteins, which is what you get in Alzheimer's and you get in Parkinson's disease and other dementias. Is you get these plaques of proteins, these large clumps of proteins inside or outside of the cells, and they interfere with sort of cellular signaling. They can also interfere with sort of the integrity of the cell walls and stuff like that. So these plaques, although you can't actually see them. So say you were to cut up somebody's brain who had dementia, these plaques, you can't see them unless you look under a microscope. You can see other things in somebody who's got dementia, so you can see that their brain is slightly smaller. The ventricles inside their brain, which are these sort of reservoirs of fluid within inside the brain, they're also shrunk as well. But these plaques, they greatly interfere with cell signaling, and the end result is cell death. So cells, the glial, the non-glial cells in the brain, they'll eventually start to die very slowly. So the typical onset of these symptoms doesn't really occur until about 10 to 15 years after they've been exposed to the protein. So these people in Papua New Guinea, they were eating the brains, but then 10 to 15 years later, they were developing these symptoms and they were dying. within a matter of years, maybe 1 or 2 years later, they were dying after showing symptoms. And the same is the case in the UK with mad cow disease. People were eating the beef, and then a few years, I think 8 years later. About 8 years later, these people were developing these symptoms and then dying. And something quite scary, actually, that one of our lecturers told us about this when we were getting this lecture was that it's quite dependent on genetics who develops symptoms and who doesn't. And he said it's quite possible that there's a genetic phenotype, so a type of people that have a specific gene that might not develop the symptoms for a long, long time after they've eaten it. So, there's still people walking around today who might have this very, very slow manifestation of the build-up of these proteins, but they don't know it yet, and then they'll eventually develop symptoms, which I thought was really, really scary. So yeah, that's kind of the sort of thing we're looking at here is something that's not alive. It's just a protein, and that's what's scary about it. That's why it's sort of almost feared in like pathology labs and in things like organ donation. That is very highly feared. They test for it rigorously because this protein's not only and also in things like neurosurgery, quite often the equipment isn't autoclaved, so it's not cleaned, it's just thrown away after a surgery because they can't confirm whether the person had CJD or not, and just washing the equipment or irradiating the equipment doesn't destroy these particles. So these particles can live on there for years and years and years without being destroyed. There's even high heat won't destroy it or get rid of all the particles, and it only takes one particle to start that chain reaction. So that it's highly feared within hospitals, although it's not a very big risk, which is, that should kind of like be a testament to how scary these things are. So, I mean, there's not much research on it because only about, I think off the top of my head, only about 100 people, only about up to 100 people in Europe, I think, are diagnosed with it every year. So it's like incredibly rare. And so, these things that are quite rare, they don't often get the research attention that they need to be sort of like dealt with. Although I suspect that there's not very much you can do once you've been infected with it. Just like dementia, there's no cure. It is fatal eventually. It might take a while. And I guess that's also where I come to the next part of this sort of like talk, is another prion disease, but a different type of prion disease called fatal familial insomnia. And this is the stuff of nightmares. So this particular prion disease is not caused by an infection, but it's caused by a genetic sort of phenotype. So only people who have this genetic phenotype will develop fatal familial insomnia. And I believe it's recessive, so you'd have to carry both alleles to present, and then even then it's not a definite, but I'd have to check that. I've only just thought of this. I would have added it into my research, but I've only just thought of this particular one. But fatal familial insomnia is where sort of middle age you start to develop symptoms of inability to go to sleep, and it's pervasive. There's absolutely no way of getting to sleep. So drugs don't work. So if these people are given sedatives to fall asleep or put into sort of like a general anesthesia, they go into a coma and they can die from it. So that doesn't work. These people are essentially condemned to die through never being able to sleep. and they do die. They die about a year to 18 months later after presenting. I think this is the scariest one, because, you know, the others are pretty scary, but they can still sleep. I think sleep is such an important part of our biology that, you know, being deprived of that for a whole year before you die would just be absolute hell. Luckily, it is also incredibly rare. It's a little bit like Huntington's disease, and it's sort of like, I don't know if you know much about Huntington's disease, but it's the sort of thing where if one of your parents has it and one of your parents doesn't have it, then you have a chance of having it, but you don't necessarily present with it, but you can be a carrier. And Huntington's disease is very similar to this particular prion disease, in that Huntington's disease is a dementia as well, and it's caused by these plaques of proteins in your brain, particularly this. I'm trying to make this not too complicated, but in your genetics you have something called a CAG repeat, which is the 3 nucleotides in your DNA. C, A, and G in that order, and then it's CAG, 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 CAG. And if you have 23 or more, or 28 or more of these CAG repeats, then you're highly likely to present with symptoms of Huntington's disease when you're older. And then any more than, I believe, 100 of these repeats, and you'll present with Huntington's disease in your sort of adolescence or late childhood, which is very scary. But these things are all determined by genetics, so they're passed down through families. Although Huntington's disease, I believe that recently they found evidence that you can actually develop more and more CAG repeats within your DNA over time, so you can actually have Huntington's disease develop within your family over generations, which is not great. So yeah, I mean, have you got any questions for any of that? I realise I literally just spoke for about 15 minutes non-stop, so I mean, go ahead with any questions you have.
Yeah, I was just so keen to let you say your piece, and I desperately didn't want to interrupt you, but there was a few things that cropped up in my mind. One of the first things was signalling, and I'm wondering, what is the signalling like that would communicate? So you've got one misfolded protein, the only one, for example, and then it comes across a normal protein that it could, if you like, convert to its misfolded shape. What is the signalling mechanism that communicates with one protein that's misfolded with a normal protein to cause that to misfold?
Sure. In this particular case, there's hundreds of different ways that proteins can interact with one another and how they actually interact. So you've got normal ways of them interacting, so they can just bump into one another, which just sort of happens by chance. And then in this particular case, so having a protein that's misfolded and a normal protein, the misfolded protein would have like an interaction, a region of interaction on the outside of that protein that would fit the shape of the correct protein. And then so the correct protein would fit into this protein, a conformational change based on, so something that's worth mentioning here is that proteins have different amino acids at different parts in the chain, and the folding gives it its function, but those amino acids kind of like act like a little bit like modules for doing stuff. So you can have proteins, amino acids that will cause chemical reactions on other molecules, and sort of like a very complex array of these things can have very sort of can cause like conformational changes in the protein. So this misfolded protein would attach to this other protein. Some of the amino acids might do something onto that other protein to cause it to change shape, and then as a result, that's now joined together and formed like a mini plaque, I guess. example of this. So something called a tyrosine kinase receptor. So it's a receptor on a cell. It has two parts to it in the cell membrane. So two parts sticking up through the cell membrane. And when the right protein comes along and attaches to it, there's an enzyme activity on the tyrosine of this protein that causes it to send another message. I'm not so great, I've had to think of this off the top of my head, I'm not so great on some of the specifics, but I know it's to do with this enzymatic activity of the tyrosine of the protein, and that causes the action of the receptor. So it might send off a secondary messenger within the cell. It might cause like a influx of certain ions into the cell. And there's others as well. There's these G protein coupled to receptors. I don't know if you've ever heard of those. Those are pretty common. They're the most abundant receptors in our brain. These GPCRs, and so they have. Oh, let me get this right. They have 7 transmembrane. So 7 parts of the protein that go in and out of the membrane of the cell, but they form a circle. So when the correct, it's almost like a cylinder of cylinders, because they're sort of like coiled into strips. And I guess I could actually find a GPCR picture to help. To help sort of illustrate what I'm trying to say here. Okay. Okay, this one should do fine. Okay, can you see my screen?
I can't, but what you might do is send me those pictures, and what I'll do is I'll include them on the video. Just send me a couple of illustrations.
Okay, yeah. No, that's fine. So these 7 cylinders of the protein are sort of conformed into like a circle, like a cylinder. So there's an opening in the middle, and what might happen is a protein or a molecule. Maybe it's a drug, like for instance, cannabis uses this particular mechanism. So the molecule of the drug will come down. It will join onto the outer membrane. The exogenous part of the protein. So this receptor site where the molecule will join in, and then that will cause like a chemical reaction within the protein to change the conformations, to change the shape of this protein. And in this instance, it will twist certain parts of the transmembrane part of the protein, and then on the inside of the cell, there's these G proteins. So they have a G alpha, a G beta of the protein. And either the G alpha will go off, or the G beta will go off, or they'll both go off, stuck together, and they'll do whatever they need to do within the cell. So that's one way that a protein might influence one another. And in the particular case of prions, I do believe it is just a simple sort of like they bump into one another, they find one another. These proteins, the PRP protein, I suspect is quite abundant in certain areas of the brain, particularly like the thalamus, the middle of the brain, which is where a lot of these prions like to start their journey. So yeah, that's the answer to that part of your question. Well, was there another question you had?
Just want to get it straight in my mind that I'm understanding that point. One thing that confused me slightly is that I thought we were talking about one misfolded protein bumping into a normal protein and changing it through the method that you've said. But what confused me is that you've mentioned a cell. So I thought they were just proteins bumping into each other, and in one of your examples, you said the receptors are attached to, if you like, the cell.
Yeah.
And yeah, go on.
Sorry, I should have clarified. The examples I gave you, so the tyrosine kinase receptor and the GPCR receptor, they're not necessarily, they're not really to do with prions. That is more of an example of how a protein might influence another protein, or how a molecule might influence another molecule, another protein. Sorry. So although prions do influence receptors, it wouldn't necessarily be in this way.
So are we, when you talk specifically about, if you like, prion disease or CJD, a misfolded protein is bumping into another protein, but where does a cell come into this?
So eventually, these misfolded proteins will become plaques, which is I guess an industry name for like a collection, like a clump of these proteins. These clumps get too big, essentially. In between the cells, there's a bit of space, but there's not much space. You don't want too much space, because then it becomes inefficient. So there's not much space, but these plaques take up too much space, so they become too big, and they get in the way. So for a brain or any tissue really to function the way it should, that this intracellular space between the cells, it needs to be sort of free flowing, so that other cells can do their work. So, for instance, one job for a particular type of cell called an astrocyte, which is not a neuron, but it's a non-glial cell. It is a glial cell. It is a glial cell. It provides structure to your brain. It also does things like hoovers up any molecules that it deems shouldn't be there. It also has a role in sort of signaling to other cells around it. It has immune system functions as well. Although microglia, which is another type of glial cell, is more responsible for immune response, but that's unrelated. So the astrocytes can't do their work. They can't suck up particular molecules or proteins in the intracellular space, or extracellular space. Signaling between cells is interrupted. So if you have, say you want to, with an example of serotonin signaling, which serotonin is pretty abundant throughout the whole brain, the serotonin needs to be released from one cell through the synapse to another cell. And if there's a big plaque in the way, that signaling is interrupted. Sometimes the plaques can also dislodge the synapses, so the synapse doesn't actually meet the synaptic bouton, which is the little end of the synapse, might be pushed away from the receiver part of the synapse, that site. So there's a lot of ways in which it can mess things up. Those are just a few. And then eventually these plaques little bits will break off, and they'll cause more, almost a bit like a cancer, like a metastatic cancer. So little bits might break off, and they might start reacting with other PRP proteins in other parts of the brain. And eventually you'll get this sort of like almost Swiss cheese consistency to the brain, like very spongy. I believe it's called something spongiform encephalopathy.
It was BSE, wasn't it?
Transmissible spongiform encephalopathy, I believe, is what it's called.
But when it was Creutzfeldt-Jakob, but it was the bovines, the cows. It was bovine, wasn't it? Spongiform encephalitis. Yeah. BSE. They used. I'm old enough to remember BSE, so I'm guessing. It's sort of,
which is the technical name for the mad cow disease.
Right.
You're right. And that's where the spongiform bit comes from. Is that it essentially turns your brain to a sponge, in a very sort of almost blunt way of saying it. It's transmissible, and it's an encephalopathy. So encephal being brain, pathy meaning sort of diseased state. So a transmissible, spongy brain diseased state.
Right.
It's really, really quite horrible.
I'm trying not to feel like that right now, but I think you've explained it clearly enough. But I just want to double check that I've got the understanding. So you've got two bits of protein. One's misfolded, one's not misfolded. They bump into each other, and then there's some receptors on the normal bit of protein that attach to this misfolded protein, and there's some signaling that happens which causes the normal protein to misfold. Is that roughly right?
Essentially, yeah, sort of. There aren't really any receptors on the protein itself. There's just a site for joining. So there's like a site that has a specific shape, and only the right shape will fit in there. And it just so happens that the right shape is this correctly folded protein.
Right. So the correctly folded protein has the right shape that matches the misfolded protein.
Yeah.
Right. And whereas if it bumped into a normal protein, is it PRP, that attachment wouldn't happen?
So if two PRPs, the normal PRP versions, if they both bumped into one another, I suspect not much would happen. I don't know that for sure, but they wouldn't join together and they wouldn't form a plaque. But the misfolded version has a site on it that will join to a correctly folded version.
I'm really sorry for drilling down on this. I don't know, I'm trying to drill down into your knowledge as well, because obviously everyone has their limits, and I'm just trying to drill down and trying to find out, is it a like you say, like a jigsaw, but is there any transmission of DNA, RNA between the two jigsaw parts, if you want to call it that? Is there an electrical, chemical sort of like, I don't know, bonding happening? Do you know what the physical method of transfer or the actual, can you drill down any further into the actual mechanism of
I can sort of, the thing is, we don't know for sure.
Okay.
Exactly how. There's no genetic transfer. I mean, proteins don't have genes themselves. They don't have any DNA. They're not big enough to really have that.
Oh, so you're saying that
dead.
You're saying the prions don't have DNA?
No, they're just proteins. They're just dead.
They're just amino acids that have been
just amino acids
that have been they're in a specific 3D shape or folded shape.
Yeah.
Okay.
That's why I think they're so fascinating. Is that it's not even a virus or a bacteria. It's not alive. It's not a parasite.
So would you?
It doesn't.
Yeah. So just I'm cutting across you here because I know you're going to have your own limits on the amount you can speak, and what I'm doing now is I'm sort of injecting some ideas and some sort of a bit of chopping you up a little bit. And in the sense that I want to try and get you into this sort of sphere of saying, would a prion, would this mysterious sort of protein that we don't know too much about, would it be less alive than a virus? Would you say, from what you said?
I mean, there's a strong case for viruses being alive.
Okay.
One that they replicate a bit like a living organism, so they I say a bit like, it's not really like bacteria or anything like that. Bacteria multiply, they sort of like copy genetic material and then they multiply, whereas a virus will get into a cell and then use the cell's sort of DNA transcribing mechanisms, all of its parenchyma, all of its machinery that it uses to make its own sort of reproduction. It uses that to create its own like offspring, essentially, but it injects the DNA. So viruses have their own. It's not DNA. Sometimes it can be DNA. Sometimes it's RNA. So they have their own genetic material, whereas these proteins don't have that. They are, I'd say, they are the true sense of dead. They're not alive at all. Whereas viruses, you could argue either way, they're alive, they're dead. but yeah, prions are 100%
Right.
not live.
I'm going to hit you with a curveball. Do you have any ideas of the evolution of viruses? Say, like if life happened 4.5 billion years ago, do you know what life? What was the evolutionary sort of trajectory of viruses? What could they have developed from?
This is really not my area of expertise.
No problem. So
However,
go on.
I'll give you I think what I suspect probably happened is that sort of organic compounds that contain carbon and oxygen and hydrogen and maybe a couple of others like nitrogen. These organic compounds were formed in like a very, very hot environment and in water. And just through some sort of miracle of statistics or miracle of probability, they may have joined together or they reacted with others. And eventually one day they built these chains of RNA of these nucleotides, which is what we've got now. And this RNA maybe recruited or found itself within so the outside of a virus is called a capsid, maybe found itself in a very primitive form of capsid, and this eventually evolved a way of being able to attack bacteria, because a lot of viruses attack bacteria, so they're called bacteriophages. So they maybe evolved the ability to enter bacteria, and then eventually evolved the ability to use that bacteria's sort of genetic transcribing machinery to make its own virus version, and then it's kind of gone on from there. So I don't know how true all of that is. I suspect that I've got a lot of that wrong, but unfortunately, viruses are just not really my expertise.
Right.
The only experience I have with viruses are learning how to use them to genetically modify things. So like mice.
Genetically modify what? Sorry.
Mice.
You've used viruses to genetically modify mice.
I personally haven't, but I've learned how it's done. So I know roughly how it's done.
Maybe that could be a subject of another future meeting. But I just want to.
It's gonna have to be.
I feel we're drawing to the end of this, but I want to dumb it down slightly, without making it trite. I just want to bring it back to the beginning where you're talking about that tribe, and you talked about there was a period of latency before things developed, say 10 years. But again, that's from what you've said. Later, that could be dependent on genetics. And this tribe, I'm just trying to picture what happened. So, say one of their elders died, and you talked about, is it case 0? Is that correct, or something like that? Case 0?
Yeah, they suspect there was a patient 0.
So, a patient 0, let's say a patient 0 in this tribe, and this man was eaten after he died, and then 10 years down the line, lots of members of this tribe then developed CJD. Was all that? One, is that roughly true? What I've remembered from what you've said, and two, was this tribe? Did it become self-limiting this case, and did a certain amount of the tribe die off? And
Well, I believe as of the 60s, they've actually stopped eating they've stopped eating the brain of the family that die, or the members of the group that die.
Right, okay.
I don't actually know if, I suspect there's not really been very much follow-up on it. This was all quite a while ago. A lot more of the research is into the more like common presentation of it, so CJD and from the BSc that you were talking about earlier.
And again, sort of trite question, and I'm aware I'm cutting across you, but I just feel I have to. Would you rather have, say, a long COVID, or would you rather have a prion disease?
Well,
Long COVID. So
yeah, I mean, I'm really not exaggerating when I say prion diseases are nightmare fuel.
Okay.
Yeah, they really are. I mean, I've worked in nursing home. I worked in nursing homes for about 5 years, sorry. And a few of these years were spent in dementia plus nursing homes, and dementia is no joke. It's like quite awful, really. I think it's one of nature's cruel jokes, essentially. Or not jokes, but it's one of nature's cruel sort of tricks that it plays on us. Viruses are bad. Things like cancer are awful, but dementia robs you of everything. Like absolutely robs you of everything and then kills you. It's quite brutal, really. And the one scary thing about dementia is, as soon as you start showing symptoms, it's too late. There's no way of easing it quite as much. Like you can treat some of the symptoms, but as soon as you start presenting symptoms, it's just a downhill from there until you die.
Okay.
So the actual pathology, so the etiology of the dementia starts up to 20 years before you even show signs or show symptoms. So by that point, the damage is done, and you get a lot of like news reports and articles saying, are we close to a cure for dementia? I think the most effective cure would simply to be prevention. So, say a vaccine. So yeah.
Okay, I'm glad you ended it on a slight upper there, because it was going, terrible, worst ever, and then prevention. Okay, so we're going to cut it short of that. But I think both you and me, I feel like I've enjoyed it. Have you enjoyed the?
Yeah, for sure.
Yeah.
I mean, I love talking about this kind of stuff.
Yeah.
I don't get to talk about it often.
No.
I don't actually like talking about it unless somebody asks specifically.
Yeah.
I think when I was going through uni, I was kind of thought I was the big dog, doing this massive degree and knowing a lot about brains, and I'd try and show off a little bit. But I think I've tried to humble myself a little bit more as of recent, and I'll only really tell people if they ask for it. So
yeah.
And I think a lot of people find this kind of stuff interesting. So I don't go around saying, I know a lot about brains, and then ask me a question. But I think if somebody genuinely wants to know, and if it's something I know about, then I'll give it a good go at explaining. But
brilliant, Archie. I want to thank you for your time. Yeah, and we're gonna end it there. And I'd say to any viewers that are still with us, thank you very much, and I'll see you in my next video. And I hope Archie's going to be back with us at some point to carry on different lines of questioning, and we're going to draw knowledge that Archie is willing to share with us, and maybe you'd be so kind to send us in the next few days just one or two pictures that you'd refer to in this in this little video.
Sure.
Okay, Archie, thanks a million, and take care.
No worries.
Stay safe.
Yeah, you too.
Take care.
Cheers. Bye.
JC out.